Depriving deadly brain tumors of cholesterol may be their Achilles heel. Adult brain cancers are almost universally fatal. This is because of the biochemical composition of the central nervous system, CNS, and the blood-brain barrier. The blood-brain barrier selectively and protectively limits the passage of molecules from the blood into the brain, and also blocks most existing chemotherapies, contributing to treatment failure. Glioblastoma multiform are extremely difficult to treat. The median survival rate is just over 14 months, with only few treated patients living 5 years or more past diagnosis. Glioblastoma multiform are among the most genomically well-characterized forms of cancer, with clear evidence of oncogene mutations. An alternative approach proves promising against this hard-to-treat cancer. Researchers at University of California San Diego School of Medicine, Ludwig Institute for Cancer Research and the Scripps Research Institute, in Los Angeles and Japan, report that depriving deadly brain cancer cells of cholesterol, which they import from neighboring healthy cells, specifically kills tumor cells and causes tumor regression and prolonged survival. This is a potential alternative method for treating glioblastomas, the most common and the most aggressive form of brain cancer. This includes blocking small molecule inhibitors that target growth factor receptors, which have not proven to be effective with brain cancers, possibly due to their inability to get past the blood-brain barrier and achieve sufficiently high levels in the central nervous system. But this information has yet to benefit patients, because the drugs designed to target these oncogenes have difficulty accessing their targets in the brain. So an alternative way to use this information has to be developed for more effective treatments. One such approach stems from the observation that oncogenes or mutated genes can rewire the biochemical pathways of cells in ways that make them dependent on proteins that are not themselves encoded by oncogenes. Targeting these oncogene-induced co-dependencies opens up a much broader pharmacopoeia, including the use of drugs that aren't traditionally part of cancer drug pipelines but have better pharmacological properties. Paul S. Mischel, M.D., a member of the Ludwig Cancer Research branch at UC San Diego and professor in the UC San Diego School of Medicine Department of Pathology, is the senior author. The findings are published in journal Cancer Cell, on October 13, 2016. In previous research, Mischel and others had noted GBM cells cannot synthesize cholesterol, which is vital to cell structure and function, particularly in the brain. Instead, GBM cells derive what they need from brain cells called astrocytes, which produce cholesterol in abundance. Roughly 20% of total body cholesterol is found in the brain. When normal cells have sufficient cholesterol, they convert some of it into molecules called oxysterols, which activates receptor in the cell's nucleus, liver X receptor, LXR, to shut down the uptake of cholesterol. So when normal cells get enough cholesterol, they stop making it, stop taking it up and start pumping it out. In GBM cells, this mechanism is completely disrupted. They're like parasites of the brain's normal cholesterol system. They steal cholesterol and don't have an off switch. They just keep gobbling the stuff up. GBM cells ensure their cholesterol supply by suppressing the production of oxysterols, ensuring cells' LXRs remain inactive. LXR 623 easily crossed the blood-brain barrier to bind with LXRs in normal cells, stimulating the production of oxysterols and the reduction of cholesterol. There was no effect upon healthy neurons and other brain cells, but GBM cells were deprived of vital cholesterol, resulting in cell death and tumor regression. Disrupting cholesterol import by GBM cells caused dramatic cancer cell death and shrinkage of the tumors significantly, prolonging the survival. The strategy worked with every single GBM tumor and even on other types of the tumors that had metastasized to the brain. LXR 623 also had minimal effect on astrocytes or other tissues of the body. The GBM strategy could be implemented in using drug candidates under development or in early trials. Thank you.